Good evening, sir. I'll be presenting on principle and instruments of gas chromatography. Gas chromatography is an analytical technique used to separate volatile components. Gas chromatography helps to separate mixtures into individual components so that each component can be identified and measured separately. It works by separating the components between a gas phase and a stationary phase. Different components move at different speeds, so separation takes place. A gas chromatography is widely used in pharmaceutical, environmental analysis, food testing, and research laboratories. Now, if we observe the image in this slide, it shows how the sample mixture enters the gas chromatography system and gets separated into individual components inside the column. Why do we need gas chromatography? We need gas chromatography because many chemical samples contain complex mixtures. GC helps to separate those mixtures into individual components. It also helps in identifying unknown chemical substance. Another important use of determining the purity and concentration of components. Gas chromatography provides rapid analysis, accurate results, and high sensitivity. This is why it is widely used in quality control and research laboratories. The image shown here represents how different chemical compounds are separated and analyzed individually. The principle of gas chromatography is based on the separation between two phases, the mobile phase and the stationary phase. The mobile phase is an inner carrier gas such as helium or nitrogen. The stationary phase is a liquid or solid present inside the column. Different components interact differently with the stationary phase. The components with lower attraction move faster and elute earlier. The components with stronger attraction stay longer and come out later. Separation mainly depends on boiling point, polarity, and affinity towards the stationary phase. If you observe this diagram, different components move through column at different speeds. Now let us see the basic working of gas chromatography. First, the sample is injected into the injector port and the sample is then vaporized instantly due to high temperature. Next, the carrier gas carries the vaporized sample into the column. Inside the column, different components separate based on their properties. After separation, the detector detects each component one by one. Finally, the results are recorded in the form of a chromatogram. This figure shows step-by-step working of gas chromatography from sample injection up to chromatogram formation. The main components of GC instrument. The main component of gas chromatography instrument are the carrier gas system, sample injection system, GC column, column oven, detector and data recording system, and each component performs a specific function. The carrier gas system acts as the mobile phase. Its main function is to carry the vaporized sample through the column. Common gases used are helium, nitrogen, hydrogen, and argon. The carrier gas should be inert, dry, and highly pure. Proper flow rate is very important for good separation. For example, if the gas flows too fast, the components may not separate properly. The sample injection system introduces the sample into the instrument. The injector temperature should vaporize the sample quickly. There are different types of injection methods. Split injection, only small amount of sample enters the column. Spitless injection, almost entire sample enters the column. On column injection, the sample is directly introduced into the column. Different methods are selected depending upon the sample type. The GC column is the most important part because the separation occurs here. There are two types of columns, packed columns and capillary columns. Packed columns contain solid support coated with stationary phase. They are less efficient. Capillary column are narrow columns with stationary phase coated on the inner wall. They provide better resolution, better efficiency, and faster analysis. This is why capillary columns are more commonly used today. These images compare the packed column and capillary columns. Capillary columns are narrower and provide better separation and efficiency compared to packed columns. The column oven maintains the required temperature. Temperature control is very important for proper separation. Temperature programming helps in separating complex mixtures more effectively. Higher temperatures reduce the retention time, so components come out faster. For example, in petroleum analysis, the temperature programming helps in separating components with different boiling points. After separation, the compounds enter the detector. The detector converts the compounds into electrical signals. The common detectors used are thermal conductivity detector or TCD, flame ionized detector or FID, electron capture detector or ECD, and mass spectrometry detector or GCMS. Different detectors are used in different applications. FID is most commonly used in detector in gas chromatography. It detects ions formed during combustion when organic compounds are burned in the flame. Ions are produced and measured as electrical signals. FID is highly sensitive for organic compounds and hydrocarbons, therefore, it is widely used in hydrocarbon analysis. Thermal conductivity detector measures the change in thermal conductivity. It is called universal detector because it can detect many compounds. It is suitable for inorganic and permanent gases. However, compared to FID, it is less sensitive. The data recording system converts the detector signals into readable chromatogram. The functions include retrieving electrical signals, amplifying weak signals, converting the signals into digital form, displaying chromatograms, calculating retention time and peak area, and storing results. This helps in quantitative and qualitative analysis. A chromatogram is a graphical output obtained from the GC instrument. It shows the detector response versus the time. The X represents the retention time and Y represents the detector response or the signal intensity. Each peak represents a different compound. The position of the peak helps to identify the compound while the peak area indicates the concentration. If we observe the chromatogram, each peak represents a different compound and the position of the peak gives the retention time while the peak area indicates the concentration of the compound. Thank you.